H5N1, highly pathogenic avian influenza, has caused infections in poultry in more than 50 countries and is a severe threat to poultry production, national economies, and to human health. This video is about using vaccination as a tool for the control of H5N1 avian influenza. Agriculture and veterinary officials may want to consider this information as they contribute to the decision of whether or not to use vaccination.
Vaccination can help reduce disease spread in poultry and thereby the risk of human exposure. However, if vaccination programs are not properly carried out, they may make it more difficult to eliminate the virus from a country. The decision to use vaccination as an aid in the control of H5N1 avian influenza is complex and involves multiple factors that are unique to each country. It is very important that government officials consider all of these factors before deciding to use vaccination in poultry as an aid to control this devastating disease. The World Organization for Animal Health, OIE, recommends that highly pathogenic avian influenza be eradicated wherever it occurs in poultry in order to protect animal health and public health. The ability to eradicate highly pathogenic avian influenza depends on the epidemiological situation in the country and on the resources and infrastructure available.
Vaccination alone will not eliminate influenza from a country, and if used, should be part of a larger control strategy that includes biosecurity, surveillance, animal movement control, and producer and consumer education. This disease is different from other poultry diseases, and designing and implementing an effective vaccination program is more complex. This virus can easily adapt and change, making it unpredictable and potentially dangerous. Influenza A viruses are named by their hemagglutinin, or H protein, and their neuraminidase, or N protein. The H protein is used by the virus to get into host cells, and the N protein is used by the virus to escape from cells to infect other cells. There are 16 different H types and 9 different N types, and any combination of the two can occur. The only animals in which all 16 H types and all 9 N types have been found are wild waterfowl and shorebirds. These birds are the reservoir for avian influenza viruses. Influenza viruses generally do not cause clinical signs in the carrier birds. All influenza viruses in other species originally emerged from birds. The influenza transmission pattern among wild waterfowl, poultry, pigs, and people is complex. In recent years, we have come to understand much more about how influenza can spread and adapt. The avian influenza virus is an RNA virus made up of 8 RNA strands. This RNA provides the blueprint for all of the proteins made by the virus, including the H protein and the N protein. In response to infection or vaccination, the host produces antibodies which attach to the H protein and block the virus from infecting a cell. Antibody to H protein is important for protection from influenza disease. A problem arises when the virus mutates and changes its H protein. Two terms used to describe changes in influenza virus H proteins are antigenic drift and antigenic shift. Antigenic drift occurs when minor changes in the RNA lead to small changes in the H protein. For example, when the H1 protein drifts, it is still an H1. However, antibody cannot bind as well to the drifted protein. This process occurs continually in influenza viruses and contributes to the seasonal influenza epidemics in people. Because of this, new human influenza vaccines are developed each year. Antigenic shift is a more significant change than antigenic drift and results in a different H type. An example of a shift would be H1 to H3 or H3 to H5. Antibody to H1 does not bind to H3. Antigenic shift can be caused by adaptive mutation. This occurs when the influenza virus jumps from one species to another and adapts to the new species. This is likely what occurred in the 1918 human pandemic that caused 40 to 50 million deaths worldwide. Antigenic shift can also be caused by genetic reassortment. This occurs when two different influenza viruses mix within a cell and a new hybrid virus emerges with a new H-type. In poultry, the terms low pathogenic and highly pathogenic are used to describe influenza viruses. Low pathogenic influenza viruses cause a mild respiratory and gastrointestinal disease in poultry. Highly pathogenic infections occur less often and cause a severe fatal disease. Only the H5 and H7 subtypes of avian influenza have caused highly pathogenic disease in poultry. Variations in the H protein influence whether the virus will be low pathogenic or highly pathogenic in poultry. Low pathogenic H5 and H7 viruses can change and become highly pathogenic. The H5N1 strain now circulating in Asia, Europe, and Africa is highly pathogenic and has resulted in the deaths of hundreds of millions of birds. This virus is considered to be the most significant potential zoonotic disease threat to human health. Infection in humans is still rare and requires very close contact with infected birds. However, scientists are very concerned that this strain of avian influenza may adapt to humans or reassort with a human influenza virus. H5N1 in humans is the result of an antigenic shift, and if the virus becomes easily transmitted between people, a worldwide influenza pandemic could occur. There have been 3 human influenza pandemics in the last 100 years, all caused by antigenic shift. The first pandemic, in 1918, occurred when an H1N1 influenza virus was transmitted from birds directly to people. This is known as the Great Pandemic. The next pandemics occurred in 1957 when the H2 and 1968 when the H3 influenza viruses were first transferred from animals to humans. Since humans didn't have immunity to these viruses, influenza pandemics occurred. But these pandemics were not nearly as severe as the pandemic in 1918. The characteristics of the current H5N1 strain suggest that an H5N1 pandemic could be as severe as the 1918 pandemic. Humans do not have immunity to H5 influenza, and like the 1918 influenza virus, the H5N1 virus causes the most severe disease in people with healthy immune systems. This particular strain of H5N1 influenza virus has a high case fatality rate, causing the deaths of about half of the people that have become infected. Understanding the basic biology of influenza A viruses helps us to realize how important it is to eradicate avian influenza from domestic bird populations as quickly as possible. The more the virus is allowed to replicate, the more opportunity there is for antigenic drift and shift. Dr. David Swain discusses 5 important components that should be part of the plan for controlling avian influenza.
1, biosecurity, which is methods by which we include or exclude the movement of that virus on or off of farms or premises, and that could include such things as quarantine. The second one is good diagnostics and surveillance programs. That would tell us where the virus is at any one time. Third would be elimination of avian influenza infected poultry. And the fourth one would be decreasing host susceptibility to the virus. And by far the most important of these is number 5, which is education, which is educating all people involved in the poultry protection segment, from the farmers to the feed suppliers, to the government, even to the executives of companies involved in those programs. By educating them, they would know what their role is in preventing or controlling or eradicating avian influenza. We must remember in disease control that vaccination can only be viewed as a single tool in a multi-procedural strategy. If we do not include, or if we lack proper biosecurity, we don't have good diagnostics or surveillance, we don't have an education program, and we don't eliminate infected poultry, the result is an avian influenza control program that is doomed to failure before you begin.
There are three general situations in which avian influenza vaccination is used. First, during an outbreak to help eradicate the virus from the region, called emergency vaccination. Second, to prevent the virus from entering a region, called preventative vaccination. And third, to reduce prevalence of the disease when other control measures have failed and the disease is endemic, called routine vaccination. In each of these situations, the overall goals are the same.
So more specific general goals that would be seen with vaccination are prevention of infection by increasing birds' resistance to avian influenza virus. The second one would be prevention of illness and death, which is a positive economic result as well as a humane result. Third, by good immunity, we decrease avian influenza virus growth in the respiratory and the alimentary tracts. Therefore, we decrease the shed into the environment, and therefore we reduce environmental contamination. As a result of the reduction in avian influenza environmental contamination, we decrease the transmission to birds on the farm, as well as decrease the spread between farms. And the final goal, by reducing the H5N1 high-path avian influenza virus shedding, we also reduce exposure to humans and produce fewer human cases.
So vaccination can increase the bird's resistance, prevent illness and death in birds, decrease environmental contamination, decrease spread to birds and farms, and reduce exposure to humans. However, vaccinated birds can still become infected and shed the virus. This can cause a silent transmission of the disease throughout the country. One of the challenges that must be addressed to have a successful vaccination program is to detect the vaccinated birds that become infected. Often, vaccinated birds that get infected do not show clinical illness. There are a couple of ways to detect infection in a vaccinated flock. One method is to use unvaccinated sentinel birds in each flock. If the flocks become infected, the sentinel birds will become ill, allowing the infection to be detected. This allows the rapid detection of infected flocks with minimal laboratory testing. However, the drawback of this system is that the virus can replicate to high levels in the sentinels, contaminate the environment, and then spread. Another approach is to use a DIVA strategy to differentiate infected from vaccinated animals. This requires a combination of a vaccine and a diagnostic test. This strategy uses a vaccine which has the H5 to induce protection, but is missing other components of the H5N1 virus. If the vaccinated birds become infected with the H5N1 virus, they will produce antibodies against the missing component. The DIVA diagnostic test will detect antibodies to that missing component. A positive test indicates the vaccinated bird is infected with H5N1. There are currently 3 types of DIVA systems. One that uses detection of antibodies to a non-structural protein, another that uses detection of antibodies to a different N protein, and a third system that uses the fowlpox recombinant vaccine for H5N1. Vaccination can be, and has been, an effective tool to help control and eradicate highly pathogenic avian influenza. There are a number of factors that influence the complex decision on whether or not to vaccinate in your country. These include the current epidemiological situation, the impact on trade, the resources available to conduct an effective vaccination program, and funding. First, we will discuss the epidemiologic situation in the country. Veterinary services should have a good understanding of the extent of H5N1 infection in wild birds and poultry, including chickens and domestic ducks, in their country and in neighboring countries. They should also know the risk of infection and spread of influenza from wild birds into poultry and among domestic poultry. There are 2 components to epidemiologic assessment, surveillance, and risk assessment. First of all, a surveillance program is needed to understand where the infection is occurring. Surveillance is essential, but it is also laborious and expensive, so the highest risk populations should be targeted first. The minimum requirements for effective surveillance can be found in the FAO Guiding Principles for Highly Pathogenic Avian Influenza Surveillance. Veterinary authorities can implement disease awareness programs for local animal caretakers and local authorities. The involvement of these individuals is important for successful surveillance. The awareness program should outline the steps for surveillance and reporting suspicious disease. Local authorities and bird owners should understand what samples to submit, how the results will be disseminated, what the response will be if the samples are positive, and what indemnity will be paid. The second component is risk assessment. Assessment of the risk of H5N1 introduction from migratory birds, importation of poultry and poultry products, infected poultry, and illegal movements of birds should be made. Farm management and marketing systems influence the probability of poultry becoming infected with H5N1. To assist with risk assessments, the FAO has defined 4 poultry production sectors according to the level of biosecurity and method of marketing birds and products. The probability of infection is higher in sectors with lower biosecurity. Production Sector 1 includes industrial integrated systems with high biosecurity and birds and products marketed commercially. Sector 2 is commercial poultry production systems with moderate to high biosecurity and birds and products that are usually marketed commercially. Sector 3 is commercial poultry production systems with low to minimal biosecurity and birds and products entering live bird markets. And Production Sector 4 includes village or backyard production with minimal biosecurity and birds and products consumed locally. 2 additional risks for transmission of avian influenza are live poultry markets and cockfighting. Highly pathogenic avian influenza viruses have been isolated from poultry and live bird markets with and without evidence of clinical disease. The infection can spread from these markets to farms by the movement of contaminated people, poultry, cages, and transport equipment. Gaming cocks have been directly implicated in the rapid spread of H5N1 in Asia. Cockfighting involves the unregulated transport of birds across borders, sometimes from areas of high H5N1 infection to uninfected areas. Live bird markets and cockfighting are part of the lifestyle and culture in many areas of the world. Identifying ways to decrease risks while respecting cultural practices is challenging and should be addressed by agriculture and veterinary officials. Another important risk assessment to be made is the risk of transmission of avian influenza from poultry to humans. In sectors where the risk is high, such as backyard poultry production with very little biosecurity, vaccination may be one of the tools used to reduce the risk of animal infection and human exposure. Using the surveillance and risk assessment information, a country's veterinary services can map high-risk zones. These maps can be used to develop a vaccination strategy at the federal level. Within each region, the local veterinary services officials should develop their specific plans based on the federal guidelines. In summary, the epidemiologic situation in the country must be understood in order to decide not only if vaccination will be an effective tool, but how to design the most effective vaccination strategy. The export market for poultry and poultry products in the affected country is a major factor in the decision to implement vaccination for highly pathogenic avian influenza. Vaccination can impact international trade in poultry and poultry products because it can potentially hide the presence of virus in a region.
When you implement vaccination, you may find some opposition to trade from your trading partners. So if you are an exporting nation, you need to work in advance with your trading partners to establish the effects on trade and also the new rules for trade once you start vaccinating. But this needs to be done in advance. If you try to do this when you're trying to implement a vaccination program, it's going to delay the introduction of vaccination. For those countries which are using vaccine perhaps in their smallholder sector but not in their large farms, they should consider the issue of compartmentalization.
Zoning and compartmentalization are 2 different procedures recognized by the OIE for establishing subpopulations of disease-free animals in a country.
Zoning is a concept quite well known, particularly in diseases such as foot-and-mouth disease, where subpopulations within a country are separated primarily on the basis of geography. We divide them using political borders or geographical borders. We have a state or a province that may be free of the disease, while another one may have another sanitary status. Whereas compartmentalization is primarily the separation of subpopulations on the basis of biosecurity measures. It will allow for trade to continue, even if the country is not free of avian influenza. It can have compartments that are free and maintained free thanks to biosecurity without vaccination, while vaccination may be applied in other subpopulations of higher risk. The resources required can come from various sectors and quite often come from the benefited. In other words, quite a lot of the resources required for implementing compartmentalization are volunteered by the industry, which is the most seriously affected, and quite often in countries where the governments don't have the resources, the industry is quite willing to come up or to share the cost of establishing such a compartment.
The OIE Terrestrial Code outlines surveillance and biosecurity steps needed to assure trading partners that poultry are free of highly pathogenic avian influenza. The exporting country's veterinary services and stakeholders define the zone or compartment, identify animals and assure traceability, define biosecurity practices in place, and give this information to the importing country. The importing country determines whether it accepts the zone or compartment by evaluating the quality of veterinary services in the exporting country and making a risk assessment. Importing countries must have science-based assurance that highly pathogenic avian influenza is not present in the region or compartment that the exported poultry are coming from. After reviewing the epidemiologic situation and trade implications, if a decision is made to use vaccination, then agriculture and veterinary officials must be prepared to implement a vaccination plan and have secured the resources to effectively implement the plan. The vaccination plan should be tailored to the structure of federal and local government, the diverse sectors of the poultry industry, the agricultural practices, the terrain, and the specific situation in the country, including cultural practices. The plan should take into account the target species, compartments, and regions to be vaccinated. It should also include the logistics of vaccination, including training and equipping the vaccination teams, selecting and obtaining a suitable vaccine and necessary equipment, keeping records of the vaccination, and assessing the overall vaccination program. The vaccination plan should include an exit strategy, the plan to end the vaccination program.
Now, your exit strategy may be many years away, especially in endemically infected countries, but at least you have to have some sort of review points along the way to say, well, this is when we're going to review our vaccination and the need for continuing vaccination, whether we need to continue vaccinating all of our original target population or whether we can stop vaccinating certain sectors of the poultry population.
Resources that should be in place for a successful vaccination program include adequate supplies of safe and effective vaccines, trained and equipped vaccination teams, sufficient diagnostic laboratory support, a surveillance plan, legal authority for vaccination, culling, and indemnity, and a public education program. The first requirement is that adequate supplies of safe and effective vaccines must be available to rapidly vaccinate the majority of poultry in a region, compartment, or country. The vaccine must match the H-type and the specific strain of virus present in the region. It may be necessary for the veterinary services in a country to conduct trials to be sure that the vaccine will be effective in reducing virus shedding and the spread of infection. Conventional inactivated vaccines require injection of individual birds with 2 doses of vaccine several weeks apart. Vaccines take time to be produced. Each country should develop a plan to produce or purchase vaccines. The most common source of vaccine is commercial companies, but generally supplies are limited. These inactivated vaccines are often produced in eggs and can take several months to manufacture. Some newer vaccine technologies, such as genetically engineered vaccines, have a much quicker production time and can be delivered within several weeks. Vaccines may also be obtained through vaccine banks, though this is limited to emergency use. These vaccine banks have been established by various countries or groups of countries. 1 to 2 weeks is usually needed to get vaccines ready for use. Once the vaccines leave the manufacturer, they must be stored at refrigerator temperatures to maintain potency. Vaccines that are allowed to stay at room temperature or higher lose their potency. Maintaining refrigerator temperatures may be difficult in tropical countries. The procedures, equipment, and supplies to maintain the cold chain should be part of the vaccination plan. Vaccination teams must be trained on standard operating procedures for all aspects of vaccination and have adequate supplies and equipment. This includes syringes, needles, biohazard disposal supplies, personal protective equipment for the vaccinators, cleaning and disinfection supplies, and a system to record which birds are vaccinated. When vaccinating commercial flocks with moderate to high biosecurity, vaccinators should follow each farm's established standard biosecurity procedures. If there is H5N1 virus in the region, vaccination crews should wear face masks and gloves as a minimum for personal protection. Vaccination crews should change clothes and disinfect all equipment when they move from one flock to the next.
Vaccination and biosecurity go hand in hand. Vaccinators and paraveterinarians. And these, of course, are your most important people because these are the ones who will be going onto the farm to actually do the vaccination. And so this means you have a responsibility here to communicate with these people on what they're supposed to do. So they must be fully trained in vaccination. They need to understand disease prevention. The last thing we want to see is for vaccinators to be spreading disease from farm to farm. And we know this has happened before with other disease control programs. So it's critical that these people are trained in the right methods and practical methods of disease prevention when they're moving from farm to farm. It's best to use hands-on training for this group, so they're actually out there with vaccination guns and vaccinating poultry. The vaccinators need to understand what their target population is, and they also have to be aware of the importance of keeping records of the birds they've vaccinated.
The diagnostic laboratory support must be in place prior to vaccination.
The diagnostic laboratory has always played a crucial role in the diagnosis of avian influenza because there are no pathognomonic lesions or clinical signs that will distinguish avian influenza from many other poultry diseases that occurred worldwide. And therefore, the diagnosis of avian influenza requires a laboratory test.
Vaccinated poultry, unvaccinated poultry, and wild birds must be tested. The diagnostic laboratory must have valid reagents, trained laboratory personnel, and standardized assays. The diagnostic tests should comply with the OIE Manual of Standards for Diagnosis of Highly Pathogenic Avian Influenza. In addition to its important role in diagnosis of avian influenza, the diagnostic laboratory is crucial for a successful surveillance and vaccination program. Ideally, there should be diagnostic tests that can detect vaccinated birds that become infected. These birds should be destroyed to prevent silent spread of the disease. Laboratory tests are used to evaluate the vaccine program by answering these questions. Is the correct strain of virus in the vaccine? How much virus is being shed after experimental challenge of vaccinated birds? How many doses are needed for adequate protection? How well are the birds responding to the vaccine? Are the vaccination crews vaccinating 100% of the birds? The legal authority in the country must have laws in place that allow for mandatory vaccination, as well as movement controls and quarantines. Legal authority is also needed for culling of infected poultry and for payment of indemnity for birds that are destroyed as part of the control program. A carefully designed and implemented indemnity program is essential for successfully managing eradication of highly pathogenic avian influenza.
Farmers have to be paid quickly in the right amount. One cannot pay too much or too little, or the compensation becomes a problem rather than contributing to the solution.
Plans should be in place for safe destruction of infected flocks. An environmentally acceptable disposal method for the destroyed birds should be determined. People who are involved in destroying infected flocks must be trained on euthanasia procedures and have adequate personal protective equipment. Decontaminating infected farms and removing or disinfecting contaminated materials are critical to effective disease control. The impact of decontamination and disposal on the local environment should be considered and approved by environmental protection agencies. A public education program is important so that farmers and bird owners cooperate and support the vaccination, surveillance, and eradication program. Education in local communities can help to facilitate mass vaccination programs. Leaders in local communities should be engaged, as they know the conditions in the area and can mobilize local resources for vaccination and surveillance. The general public should be informed of the vaccination, surveillance, and eradication program and understand movement restriction regulations. It is also important to reassure the public that it is safe to eat healthy poultry and vaccinated poultry during an outbreak. Although cooking will kill the influenza virus, infected birds and eggs from infected birds should not be eaten. In addition to all of the factors already discussed, a plan for obtaining funding for support of the preparedness and response activities is important. Prevention and control of highly pathogenic avian influenza can be very expensive. Funds should be available to implement the plan on an emergency or long-term basis. Agriculture and veterinary officials should work with the legislative branch of the country to create laws that will support the development of an emergency response program, secure funding for the program, and seek the cooperation of other agencies within the government. These agencies may have responsibilities for wildlife, the environment, human and public health, public works, security, finance, commerce, trade, foreign affairs, and communication. A country may wish to explore several potential sources of funding to support a highly pathogenic avian influenza control program. This can include government sources, the World Bank Avian Influenza Program, regional development banks such as the Asian Development Bank, the United States Agency for International Development, the European Commission, the United Nations Food and Agriculture Organization, and private funding. The commercial poultry industry in a country should be asked to contribute resources to help control highly pathogenic avian influenza.
Controlling avian influenza is really an enormous challenge for the veterinary community and for local administrations. The ecology and epidemiology of this disease are in constant evolution as it spreads to new ecosystems and hosts. And therefore, it is unthinkable to believe that we can manage this emergency without significant funds and resources.
What can your country do to prepare for or respond to highly pathogenic avian influenza? Assess the veterinary infrastructure. The OIE is encouraging countries to use the Performance, Vision, and Strategy, or PVS, tool to assess the capabilities of their veterinary infrastructure. This tool can analyze the structure and capabilities of government veterinary services and show gaps between current performance and international standards. The results can be used to guide additional investments in veterinary infrastructure to better protect animal health, human health, and food production. Request assistance. If the H5N1 virus is in your country, it is important to understand where it is and what the risks are for spread into other areas. Do not hesitate to request international assistance. The FAO's Animal Disease Crisis Management Center in Rome will provide animal disease analysis and information, as well as deploy international resources to prevent and contain dangerous animal diseases. The OFLU network, a partnership of the OIE and the FAO, can also provide assistance. This network of avian influenza experts works to exchange scientific data and biological materials and to share this information. It also offers technical advice and veterinary expertise to assist in the diagnosis, surveillance, and control of avian influenza. Be transparent. Because of the potential consequences of avian influenza on human health and food production, transparency of the current disease situation in your country is very important. Information should be shared within the veterinary services, with other agencies of the government, and with the international community. This will enable a more coordinated response to avian influenza. Review poultry industry practices. Work with other agencies in your government to examine poultry production and distribution practices and consider modifications, including marketing systems, placing additional controls on live bird markets, such as spot checks, days of rest between market days, and cleaning requirements. Segregation of species. Separating domestic waterfowl and poultry, and pigs and poultry, during production and marketing to prevent transmission between species. Farming systems and practices. Establishing poultry-free zones around Sector 1 and 2 operations, encouraging all-in, all-out production, and following strict biosecurity procedures. Transport. Requiring cleaning and disinfection of cages for transport, and the use of plastic or metal cages rather than wood cages. Other components of this vaccine decision analysis project are available to provide more in-depth information, including social and economic factors that impact the decision to use vaccination to control avian influenza, managing control and vaccination programs for highly pathogenic avian influenza, vaccination and diagnostic testing for H5N1 avian influenza. In addition, there are 16 presentations on various topics by avian influenza experts and a panel discussion on the use of vaccination in specific circumstances. In conclusion, consider the factors discussed here and determine if vaccination is a viable option for your country. Understanding the epidemiologic situation, trade implications, and veterinary resources needed to carry out a vaccination program are important first steps to determine if vaccination is a viable option for your country. If vaccination is a tool that you want to use, develop a vaccination plan so that it will be in place when it is needed, and establish relationships and commitments to assure funds to support your efforts against highly pathogenic avian influenza.